Morning. This time I'll share protein metabolism part 1. The topic that I will share with you start from protein overview, protein synthesis, and protein breakdown. Protein is a macromolecule that play many critical roles in the body, made up of smaller unit called amino acid that attach to one another with peptide bond to create polypeptide. Let's see this one in the right side. This is a protein. This one is a representative of amino acid residue. This amino acid residue will attach one another by peptide bond. And if this protein hydrolyzes into amino acid, then this amino acid can be used to build new protein. Protein structure is the three-dimensional arrangement of carbon in amino acid chain molecule. Protein are polymer, specifically polypeptide. Polypeptide form from a sequence of amino acid, the monomer of this polymer. Protein structure range inside from 10 to several thousand of amino acid. The different level of protein structure are known as primary, secondary, tertiary, and quaternary. Protein plays many roles. For example, cell signaling receptor, signaling molecule, structure membrane, enzyme, intracellular trafficking component, extracellular matrix scaffold, ion pump, ion channel, transporter, etc. Let's see protein as a fuel source. Protein are not stored for later use, so excess protein must be converted into glucose or triglyceride to build the energy reserve. Let's see the figure on the right side. We have three macromolecules here. We have a protein, carbohydrate, and lipid. Let's start with this carbohydrate. This carbohydrate, if hydrolyzed, will convert into monosaccharides such as glucose, fructose, and galactose. This monosaccharide will enter glycolysis pathway to produce pyruvic acid, and then this pyruvic acid will convert into acetyl-CoA. Then this acetyl-CoA will be used in citric acid cycle to produce energy in the form of chemical energy such as ATP. Now let's see that if this glucose 6-phosphate in a certain condition is higher than normal, then this glucose 6-phosphate can be converted into glycogen by glycogenesis. First, if this glucose 6-phosphate lower than normal, then glycogen can be converted again into glucose 6-phosphate by using this glycogeno, glycogenolysis pathway. Then this pyruvic acid also can be converted into lactic acid. Now let's move to this lipid. Lipid consists of fatty acid and glycerol. This fatty acid will be oxidized into this fatty acid spiral. Then this fatty acid spiral will convert into acetyl-CoA. Then again, the same, this acetyl-CoA will use by citric acid cycle to produce energy. But then let's see if this acetyl-CoA abundant in the system, then this acetyl-CoA can be converted into lipid through lipogenesis. But let's see the protein. Protein is a macromolecule. If we hydrolyze this molecule, it will convert into amino acid. And this amino acid will enter the nitrogen pool. This nitrogen pool will be converted into tissue or protein, and some of it will be degraded to produce ammonia and carbon dioxide. Then this ammonia, because this is so toxic, then this will be neutralized in urea cycle. Then from this urea cycle, then it will release urea. Now let's see compare the three of this macromolecule. Two of the molecule has the ability to convert into the beginning of the pathway. But in case of discussing a protein, it doesn't. So it means that protein are not stored for a later use. So excess protein must be converted into glucose or triglyceride to build the energy reserves. Now let's move to protein metabolism, cellular view. This is a central dogma, plays a big role in protein biochemistry in human body. Then, what is central dogma? Central dogma is a process by which the instruction in DNA are converted into a functional product. It was first proposed in 1958 by Francis Crick, discovered the structure of DNA. The central dogma explains a flow of genetic information from DNA to RNA, then to make a functional product as a protein. So let's see this one. This is a process of transcription, then will continue to translation, and after that will be enter into post-translational and protein degradation sometime. Let's see this one. This is a DNA. DNA in a living matter probably will replicate first, then will convert into RNA, and this RNA will translate the instruction of a DNA into a protein. Now let's see the protein metabolism. Maintenance of body protein and amino acid homeostasis are regulated by this principal protein metabolic system. Let's see this diagram. This is amino acid. This amino acid can be found from intake or from de novo synthesis. This amino acid will be used as a starting material to produce a protein, this polymer, polypeptide. Then this protein also can be hydrolyzed to convert into amino acid by a protein breakdown. Then this amino acid will be oxidized to produce urea and carbon dioxide, and this urea will be secreted from our body. So let's see this maintenance of body protein and amino acid homeostasis like this. Now let's move to protein synthesis. This protein synthesis occur in two states, transcription and translation. Transcription is transfer of genetic instruction in DNA to mRNA in nucleus. Let's see this one. Transfer of genetic instruction in DNA to RNA in nucleus. Then let's move to translation. In this case, is reading the instruction in mRNA, and tRNA bring the correct sequence of amino acid, and rRNA form bond between the amino acid. So this is the mechanism of protein synthesis in the cell. Now let's move to transcription. This one, the initiation process of this is RNA polymerase bind to a promoter, and as an elongation, addition of nucleotide to mRNA strand. Then for termination, mRNA strand detached from a DNA. Now let's move to translation. Initiation for this one is ribosome get together with mRNA and the first tRNA. Then as an elongation, these amino acids are brought to a ribosome by tRNA. Then a termination is finished, polypeptide is released to go. Now let's discuss the protein breakdown. This protein breakdown occur in somat by pepsin and hydrochloric acid. This hydrochloric acid environment, the pH is around 1.5 to 3.5. This pH will denaturate protein in the food. Pepsin break a protein into smaller polypeptide and amino acid. The digest protein enter the small intestine like this. Then pancreas release sodium bicarbonate, secretin, and CCK to protect the lining of the intestine and stimulate digestive processes. Pancreas also releases most of digestive enzymes such as trypsin, chymotrypsin, and elastase. Now let's move to digestive enzyme. Proteolytic enzymes are secreted in its proenzyme form to protect the protein component of the pancreas. Enteropeptidase will activate trypsin, trypsin, and this trypsin act as activator for other digestive enzyme. Let's see this one. This one, enteropeptidase, will activate this trypsin, and this trypsin act as activator for other digestive enzyme, such as, let's see this one, proelastase will be activated into elastase, procarboxypeptidase will be activated into carboxypeptidase, chymotrypsinogen will be activated into chymotrypsin, and prolipase will be activated into lipase. Then let's see this enzymatic proteolytic. Digestive enzyme from pancreatic juice will break complex protein into a smaller fragment. This fragment will be transported across intestinal mucosa to create new protein or convert into fat or acetyl-CoA for a Krebs cycle. Let's see this one. This is a protein. This protein will be converted into a small polypeptide by pepsin, and this polypeptide will be converted into small peptide by this trypsin, and this polypeptide will be converted into amino acid by peptidase. So together of this enzyme break complex protein into a smaller individual amino acid, which are then transported across the intestinal mucosa to be used to create new protein, or to be converted into fat or acetyl-CoA and used in the Krebs cycle. Then let's move to amino acid pool. Once the protein have been hydrolyzed, pre-amino acid join the non-essential amino acid synthesized in the liver on the amino acid pools. See this one? Amino acid pool is used for a synthesis of protein and another nitrogen containing a compound. If the amino acids are not used, they will be degraded into urea. So let's see this one. This is a body protein. This body protein, a dietary protein. From dietary protein will be digested to convert into amino acid pool. The same thing from body protein, there will be a turnover to convert into amino acid. Then this amino acid somehow can be converted to a citric acid cycle, and some of it will be converted into urea. I think that's all for this time. Thank you so much for your attention. See you then by next time, sharing with me by discussing protein metabolism part 2. Thank you so much. See you then. Bye bye.